All right. Good morning, everyone. Can you all hear me back there? This says it's on, but I don't think it is. But you probably noticed that I don't really need a microphone. Maybe by the end of today, after all of our discussion that we have, that might not be the case, but we'll see how things go with that. And the lovely weather we have out there. Sorry for you folks that were wishing to come to, South Texas and have a nice clear day. Hopefully it'll get a little bit nicer for us. I'm Dr. George DiGiovanni. I'm with the University of Texas School of Public Health and chair of the conference here. And I'm going to just give a couple of brief introductory comments. The first thing that I want to start out with is notice that we have a pretty good sized group here, and we're glad to see everyone here. It's terrific that you all could come and participate in the conference. But we have also a great diversity of different types of scientists, folks from academia. Thank you.
Yes, sir.
And folks from regulatory agencies. So just a quick show of hands, how many of you are from a regulatory agency? Can you raise your hand up? Great. And how about university or research laboratories? Super. And how about commercial laboratories or water quality labs? I know there are a few here. Great. All right. Well, it's terrific to have everyone here. Obviously, we can't go around the room and introduce everyone, but I want you all to know that this is a fairly informal conference. It is really geared towards discussion. We do have question and answer sessions after the end of each of the themes for each part of the conference. So I want to make sure that everyone knows that they can participate in that. Feel free to make comments at that time. And we also have little white cards for you shy folks out there. Okay, so if you do have a question, but you don't really want to necessarily jump up and come up to the microphone, you can write it down on your little note card here and pass it to the ends of the table. Okay, and those can be used for the Q&A sessions. And also, more importantly, we will have a panel discussion at the end of the conference. And if you have general questions that you feel would be a good part of that panel discussion, please. I don't think it's going to stay. Yeah, that's very good. Please hand your card with your comment on it to either Brian or Courtney, and I think she's in the back there, and we'll use those for the panel discussion session, okay? So keep that in mind. You can also just go put it back on the registration desk if you're really, really shy. All right. We have restrooms down the hallway there. I think there are also some over here. We have coffee and other drinks in this back room here. That's where we'll have our breaks at. Feel free to get up and go use a restroom, go and get some coffee, whatever you need during the presentations and things. We want this to be a very comfortable atmosphere for you all. All right. The other thing that is not in your packets, and you have some great materials there that our folks prepared for you, there are a couple of informational pamphlets at the back tables, all right, back there, and they look like this, the little kind of mini pamphlets there. And they're good informational packets, summaries of why bacterial source tracking is a tool that can be used for identifying sources of fecal pollution. And for those of you that are really interested too about the cost of things, you can see some of the information about different analytical methods and some of the estimated costs associated with those. Okay? All right. At this point, we're going to go ahead and kick off with Aaron Wendt from the Texas State Soil and Water Conservation Board. There he is. Didn't see him back there. And he's going to talk about some of our conference objectives. Thank you very much, and look forward to a great conference with you all.
Thank you. Good morning. This is a pretty great crowd. I'm glad everybody made it. I'm going to kind of help us frame the next day and a half and talk about the objectives from our perspective, from my agency's perspective, from the planning committee's perspective. so we can have a really good discussion. Like Dr. Giovanni said, this is intended to be a really open forum, and we want this to be a dialogue. We don't want the speakers up here to be lecturing for a day and a half, so ask questions throughout. I think to help frame this bacterial source tracking, I want to give a very short to brief timeline of bacterial source tracking in Texas. I think that prior to my time with this agency, back in the early 2000s, TCQ, our sister agency, my agency, and then several partner groups really started utilizing bacterial source tracking in water quality efforts. Our 303D list of impaired water bodies is dominated by bacteria here in Texas, and we started exploring bacterial source tracking and developing a statewide library and looking at the different methods and experimenting with bacterial source tracking in a variety of watersheds across the state. About middle 2000s, 2006, 2007, several bacteria TMDLs across Texas caused the commission and my board to take a hard look at bacteria, total maximum daily loads, and all the different tools, modeling, bacterial source tracking, and other ways to develop those. And that task force is going to be talked about a little bit later on the agenda in more detail. After the task force concluded its work, BST continued to evolve in a whole nother group of watersheds across the state. We continued to use different approaches and continued to develop that statewide library of known sources. And that kind of brings us real quickly through over a decade of work in Texas to this conference right here. And so you might be wondering why I'm up here talking about these objectives, and I think it's because my agency provided a grant to TWRI to help sponsor and plan this conference. Maybe, I don't know, a year and a half ago we started talking about this conference and began pulling a planning committee together. I really want to thank the entire planning committee. They're on the back of the agenda document, and of course TWRI and Courtney for actually doing a lot of the work to pull this conference together. One of the most important things that we felt we needed to do at the beginning was to query our partners about what they need to know about BST, what questions continue to be raised and asked by staff, by executive management, by elected and appointed officials as far as the state of BST science. And so we queried several of our partners like EPA in Dallas and TCQ and Texas Parks and Wildlife and a few others. And we asked that planning committee, the planning committee asked that group to give us their questions and what issues we needed to talk about here at this conference. And so the agenda and the speakers are really designed around the responses we got from those agencies. And this is pretty much the text from one of the first versions of the agenda that y'all saw. This kind of boils it down, I think, to what we want to talk about, what the planning committee wants to talk about, and wants to discuss with everybody in the room. Like I said before, our 303D list continues to grow in Texas, and our water quality impairments continue to grow by huge numbers, and it continues to be dominated by bacteria. And so while there's other questions dealing with water quality standards and models and other issues, we want to talk about the use of BST as a tool to help those efforts, to help stakeholders develop solutions for their watersheds to solve water quality problems. And so, as I showed on that timeline, I think that since the mid-2000s, since that task force, a lot of work has been done by various entities across the state and nation, but we really haven't had a sit-down, a concerted effort here in Texas to really talk about what have all the partners been doing and where have we advanced the science and where do questions continue to linger. And so that's really the focus of today. And it's a broad target audience. And I think that we have a mix of just about everyone out here. We have state and federal agencies. We have state and federal agencies from across the entire nation. We have academia, and we have stakeholders here in the room. We have people in the room today that are on the ground trying to solve water quality issues in their watersheds. I think for me that sums up the objectives and what we hope to accomplish today. I don't know, does anybody have any objectives they'd like to accomplish that maybe I didn't touch on? No? Okay. Back to Dr. DiGiovanni.
Thank you, Aaron. All right, our first speaker will be Dr. Don Stoeckel, and he is with the Battelle Memorial Institute in Columbus, Ohio. He was previously with the USGS and was quite involved with source tracking work. He's doing other types of source tracking related research right now, and other microbiology related work with Battelle. And we are very happy to have him here to kick off the conference and give us an overview of source tracking, past, present, and future. So maybe we can just wrap it up at the end of that. No, I'm just kidding. No. Don is,
Keep on talking, George.
yeah, he's going to find it here.
Courtney! What do you think we
I probably would have said Joe.
What do we got? Thanks, Courtney. I got it.
You're the man. Okay, great. Take over.
Great, thanks. Good morning. You have to have a hell of a peripheral vision in this room. I'll try to, well, anyway, we'll do our best. So my charge this morning, as I understand it, is to bring everybody on the same page. And because of, I guess I have had the pleasure and the honor of working with many of the panel members for the past 10 or 12 years on source tracking. And as far as the recent history of source tracking goes, that's about as far back as it goes. I was nominated as being a person who was able to give the history of source tracking, give some of the explanations, and try to lead into some of the big ideas that I think we want to accomplish, with the big ideas that we want to address. Sorry. There will be no wardrobe malfunctions, but I can't say anything for the technology. Can everybody hear me okay? Okay, great. Jesus. How about if we do that? Is that good? Try not to fade too much in and out, and I'll try not to take too much time because I know Jody deserves some time. I want to go to the Wayback Machine and talk about really the conceptually, in terms of the overall framework, what are we trying to accomplish with tracking sources of fecal contamination? So I'm going to go way back to the 1800s, just briefly. Talk about the solutions, how the issues of fecal contamination have been addressed over time, and some of the lessons that have been learned even before the modern era, which just for benefit of a round number, we'll say started in 2000. Because there are a lot of things that can be learned from the good work that was done in the 70s, 80s, 90s. And there's a lot of information that gives us a perspective that came out of the public health microbiology of the 50s and 60s that led to the current regulatory structure. What does the current regulatory structure mean? What are we trying to accomplish with that? And how can we use modern BST tools in order to accomplish those goals, either within the context or as adjunct to the context of the current regulatory structure. Where we are and where we're going, I can speak to that, and will speak to that at the end, but I'm going to give that a little bit of light service because I think that setting the frame, setting the context is more important, and we have an exceptional group of people speaking over the next few hours who are going to bring us really up to speed of what's happened since 2000 and what's going on in the modern age. So I'm going to try to set the context and get everybody on the same page. Harwood's going to talk to us about how this is done because she's got fantastic experience with multiple types of microbial source tracking. And then the late morning, afternoon sessions you can read about in the book are going to be very, very illuminating. So we'll see how illuminating this is. People didn't, can I say this? People didn't give a shit about shit until the 1800s. It flowed down the sewers. There was night soil being dropped off of balconies left and right. It was a disgusting place to live. Pretty much the cholera epidemics of the 1800s caused enough of an issue that people said, oh, what's going on here? This is unacceptable. We can't have people dying like that. It wasn't until the classic experiments and work of the epidemiologist, before the term was coined, John Snow, and the pump handle in London, showing the link between fecal contamination and fecal oral pathogens. My point being, 1895, in the late 1800s, there's an explosion of sanitary engineering in the big cities as they're being built. The reason was because of public health microbiology. People had dealt with the stench and the muck for decades, if not a century. They couldn't deal with the public health issues that reached a peak when the cholera epidemics were coming around. They drew the link, public health microbiology, drew the link between disease and fecal contamination, and they decided something needed to be done about it. First thing was to isolate the fecal contamination, and then the second thing was to figure out how not to drink it. Yep. You all know about fecal oral pathogens. It's not the feces itself; it's the pathogens. It's not the indicators itself; it's the pathogens that they indicate. And I just want to get that out so that everybody recognizes that manure is manure. It's fine fertilizer. You just don't want it in the water. If you've ever done the experiment with grade school kids where you dilute dog feces through a series of water bottles and ask them at what level are they willing to drink it, they see the dog feces go into the first bottle. They see you dilute it twice, just 1 ml dilutions, maybe 1 to 1,000 dilutions. They're happy to drink it as soon as it doesn't stink and as soon as it's not brown. I didn't let them, but they would have been happy to drink it. You can't always see the fecal contamination. You can't always smell it. You have to have some way to know what's happening. Current regulatory structure allows us to do that. The use of fecal indicator bacteria allows us to do that. And as a result, not to downplay the importance of public health microbiology, but rather to support the importance of public health microbiology. These are outdated because these were the slides I had, but it shows the point. 2000 to 2002, 2001, 2002. How many waterborne outbreaks were there? 65 in the United States. Because we're doing a good job of controlling public health, because we're doing a pretty good job of keeping people safe with the current regulatory structure. It has its flaws like anything else. These guys will tell you we don't shy away from flaws. We seek out the flaws and we try to address them. Where are they coming from? Who knew? Swimming pools is 60% in that time frame. 60% of the outbreaks came from swimming pools. What are the diseases? You've got crypto, E. coli, Shigella, norovirus, and then unknown etiology in drinking water, in swimming pool water. In fresh water, you've got Giardia, crypto, norovirus, E. coli. Naegleria didn't hit that year, but occasionally you get a Naegleria. But you've got a couple of big players that are causing these problems. E. coli, just to reiterate the point about fecal indicator bacteria, E. coli is a workhorse of a fecal indicator bacterium. 25% of illnesses in fresh water, 5% of illnesses in treated water, although it's not. The generic E. coli, of course, are not pathogenic. Some of the E. coli are pathogens, so it gets a little confusing. Y'all are probably aware of those issues. I just wanted to bring it out. I'm going to talk a little bit about food. A lot more illnesses due to food, a lot more outbreaks. This is, again, outdated. It's 2002, but that's when I gave that interview seminar. We have 68,000 cases of confirmed etiological outbreaks in food. Half of them were bacterial, half of them were viruses. Same roster of pathogens, except, well, C. perfringens, E. coli. Yeah, there's crypto over there. Salmonella, Shigella, Staph aureus. What does food have to do with this? This is part of my central thesis, so work with me. Food is irrigated with water. Ready-to-eat foods are irrigated with water. And a substantial portion of those foodborne outbreaks are coming from finfish, shellfish, vegetables, fruits and nuts, ready-to-eat vegetables and fruits. So it's not just recreational water, it's not just drinking water, and it's not just the recreation drinking shellfish harvesting water, but it's also irrigation water that is of concern in the modern age. What's the regulatory response? Getting back to how do we know whether the water is contaminated or not? Our old friend Theodor von Escherich describes Bacillus coli in about the same time frame that we're trying to control cholera, we're trying to control fecal contamination in the cities. Methods are developed. The Durham tube, anybody who's done MPNs is probably using a Durham tube. Methods are developed 150 years ago, okay, 130 years ago, not to exaggerate, to detect fecal indicator microorganisms in water so that you can tell, even if it's not stinky, even if it doesn't taste bad, you can tell whether or not there is an unacceptable risk of pathogens in that water. Explosion in public health microbiology technologies. A lot of it is led by standard methods. A lot of it is led by EPA. We now have something better than the Durham tubes in various broths, and BG, bio-green lactose broth. We have MFC agar for fecals. We have MTEC. God, I'm glad that's gone now. We have modified MTEC to detect E. coli directly. We can detect total coliforms or E. coli on quantitrays. Microbiology made easy. So now we have both the indicators and the tools to detect those indicators. And our public health statistics show that we've got, extrapolate out, factor in how many unreported cases there are. We've got thousands or tens of thousands of people out of 300 million who get sick each year from water-originated disease. We're doing a fine job, but we still have regulations to help us maintain that and to do a better job. The regulations deal with drinking water and groundwater, recreational water and shellfish harvesting waters. Those regulations are reactive. Somebody gets sick. You know, who heard of crypto until Milwaukee? Who heard of E. coli 0157 until Walkerton? These regulations are reactive, and an example, bringing us back to food, is what the this is a little soapbox since I get to stand here in front of y'all. The leafy greens marketing agreement that followed the E. coli outbreak on spinach in 2006. The regulations, one of the points I want to bring across is that the regulations that we're operating under are meant to be science-based and they're meant to be risk-based. They're meant to be, what's our recreational water standards? Al DeFore wrote a nice chapter about those, describing where those came about. It's not that there's anything magic about 8 illnesses per 1,000. There's nothing magic about, is it 19 illnesses per 1,000 in salt water. That's the background level in the 1950s when they developed the standards. What is acceptable? If it's measurable, it's not acceptable, so that's the level at which you can start to measure an impact of recreational water quality. Those very same standards have been promulgated up from total coliforms, through fecal to E. coli, and now they're being applied to leafy greens. The point being that regulations and criteria and attempts to control public health the public health to enable public health are meant to be science and risk-based. But in application, the policymakers have a hard job to do because there's not enough science to back up the epidemiological relations in a lot of cases. My water in Ohio is not the same as your water in Texas. There are different risks. There are different sources of fecal contamination. There are different epidemiological relations. There's probably a different risk from drinking a cupful of each of those waters. And yet the policymakers have to set some level. And so the regulations end up being developed based upon achievability and acceptability. And the leafy greens document in particular is I don't want to tear it down because I think that it is very useful to have a benchmark, and that having a benchmark certainly protects public health. But applying 60-year-old standards that were developed in recreational water in the Ohio River to water that's used to irrigate spinach and saying that that's science-based is a little bit of a reach. So one of the warts is that we do not have a good solid science basis for one of these reactive regulations, which may very well be incorporated into the produce safety rule of the Farm Safety Modernization Act. However, it's a starting point. And there is value to having a starting point, and we can help control by having at least something to work with. How's that for a soapbox? One of the results is that the regulatory arena is confusing because it's not a coordinated set of regulations. The basis of the regulatory structure is in large part based on achievability and acceptability, and gut check, is this going to function or not? So we have a large number of different organisms that have been used or proposed for regulatory monitoring. The workhorse, the one that's found in nearly all of the different arenas, is E. coli, or its cousin group, the fecal coliforms. Where am I going from there? I want to point out that in terms of public health, we're trying to protect public health, and in the modern age, a little bit more, we're trying to protect environmental health. Fecal indicator bacteria do indicate the presence of pathogens. They are very useful in protecting public health, but recognize that there are some flaws. Recognize that they're not the end-all. And this comes in part because of the discussions we had prior to the meeting. I know that the regulatory folks in the room say, I've got a law that says I have to use E. coli. Well, yeah, the law does say you have to use E. coli, and you have to deal with that. But there are other aspects that are also of value, and if to the extent that you have latitude to go beyond and to go the extra mile, there are other things that can be done in addition to meeting the letter of the law. The E. coli in particular isn't going to relate to swimmer's ear or skin rashes. It's not going to help you much with toxic cyanobacteria. E. coli may, they're even in its direct paradigm of E. coli being linked to fecal indicator bacteria, it's not a perfect relation. So it's not the end-all, it's the regulatory goal, but it's in terms of meeting our underlying goals, it's not the end-all. I say this because we're going to talk about other organisms that are non-regulatory. I'm just kind of padding the audience and to make sure that, that you understand why we're going to talk about E. coli in terms of fecal source tracking, but we're also talking about other organisms that can give more reliable data about fecal sources, but less reliable data about how can you meet your regulations. So Jody Harwood is going to give an exceptional talk in just a few moments about the nuts and bolts of source tracking. I'm going to give you an introduction. I'm going to warm you up a little bit so she doesn't have to break so much new ground. I'm just going to try to take the sod off. She'll go in and do the real work. The fundamental concept is that your gut is different from a chicken's gut, is different from a cow's gut, right? Ecologically speaking, there is a relation between the microbes in your gut and your physiology, your ability to digest foods. Some of you have probably read about the linkage between the microbial flora in your gut and your weight. People like George, not to say what class he falls into, have a different flora from people like Oren. It's just you have an interaction with your gut. There is a clear evolutionary, co-evolutionary relation between the host and the microbes that are inside. And there are different, the metagenomics show that there are census of organisms, clearly different populations in the human gut compared to the cow gut, compared to the chicken gut, et cetera, because of a lot of different reasons. So what's the process? All of the methods we're talking about start with a hypothesis. Collect some information, evaluate what's in the guts of your target organisms, figure out what's different, prove that it's different, And then go out into environmental waters, and when you have fecal contamination, try to find that thing that is associated with humans or associated with cattle so that you can evaluate, well, the fecal contamination here came from humans, the fecal contamination there came from cattle, which is all awesome when you've only got one source. And we'll talk about that a little bit later. The early methods going in the Wayback Machine again, we're not going all the way back to 1890, but we are going to go back to the 1960s with the fecal coliform fecal strep work that Geldrick was one of the major authors on anyway. I don't know if he actually did it. I never met the man. But there's a nice correlation that the fecal coliform fecal strep ratio in human fecal material is different from that in most other warm-blooded animals. So it's great. If you make up two jars of poop soup, you can tell which one came from humans and which one came from another animal. The problem is that if you don't know where the poop got into the river, you don't know how long it's been in the river or lake, and then we've got differential die-off. The fecal coliforms die off faster, I think.
Depends where.
Oh, come on. All right. Whatever the case, the relation doesn't hold in aged sewage. So you have to know where it's coming from in order to know whether it's human or non-human. If you know where it's coming from, what's the point? There have been other early methods that have carried through to the modern age. The sorbitol fermenting bifidobacteria, say that 3 times fast. Rhodococcus for ruminants. I think that's an organism that's on grassy substrates, so you know whether you've got some grass-fed dairy going on. Chemical methods: human origin biochemicals, metabolites, caffeine, optical brighteners has carried through to the modern age. We didn't start in 2000. There's a history of fecal source tracking attempts, starting with what indicators can I use to show that there's any poo in the water, and carrying into, well, how do I know whether the fecal contamination is of a source that I can control? Whether it's because you want to control it or whether it's because you believe that human origin fecal contamination inherently has a higher health risk than contamination from other origins. The library-dependent methods. Just realized I can see it on the screen in front of me. That'll help. Same methodology, but we're going to apply it to the library-dependent. The library-dependent methods are the ones where you collect a library of isolates of the fecal indicator, and you go through one by one and you say, that E. coli came from Jody, that E. coli came from Mike, that E. coli came from Chuck, that E. coli came from a chicken. Cultivate those puppies out of your target feces. You use some method to characterize them. Usually it's a profile, and we'll talk about some of the different profiling methods in a few minutes. You've got that library of profiles from, let's just say we're trying to do human from non-human. You've got human sources, you've got alternate sources, you do magic of statistics, or you do direct matching to see what they look like. When you cultivate organisms from the environment, you run your matching algorithm, whether it's a nearest neighbor one-to-one match or if it's a discriminant analysis to allow it to cluster. And so you're going to classify your E. coli directly, one-to-one. If you isolate 120 E. coli, if you isolate 100 E. coli and 30 of them come from humans, well, you got 30% human contamination. That makes your interpretation much easier. Some of the patterns that we're looking at that have been used traditionally, some of them are still in use, some of them have fallen out. Antibiotic resistance, carbon utilization, GC-FAME, fatty acid patterns, rep-PCR, PFGE, ribotyping, multi-locus sequence typing. These are all things that you may have heard of from your past work. What do you use, George, right now?
Eric and ribotyping.
Okay, so we've got the ERIC version of rep-PCR and ribotyping used in the state. I think Joanna does some antibiotic resistance analysis. So these are things that are currently used in the state of Texas. The magic of statistics, thanks to Mike for putting this fantastic example plot in one of his early publications. This is rep-PCR fingerprints. And by doing multivariate analysis on those fingerprints, you can distill it down into 2 axes and say, well, The goose fingerprints in general look like this. The pig fingerprints in general are distinct from the goose fingerprints. If I have an isolate from the environment and I put the same classification rules on that isolate and it falls here, I'm going to call it a pig. At that, 2000 was really this was 2000, wasn't it? Yeah. It was really an important year in microbial source tracking. There were great advances made, and we all put up our victory signs at some level. And then we discovered, well, that's worked great in the calibration data set, and when we went to validate, there are some issues. So I'll show you some examples. I'll go ahead and disclose the warts, but this is the goal, that when you have multivariate, I think this was, what was this, Mike? MANOVA. When you do a MANOVA analysis or a discriminant analysis or even a PCA, you're trying to come up with a classification rule. It's a modeling methodology. Library-independent methods, also known as marker-based methods. You're not looking for a profile, you're looking for one host-associated marker that's going to say, oh yeah, I recognize this is chicken. You're not looking for a pattern, you're looking for one thing. Instead of isolating organisms from the fecal material from your reference feces, you're still collecting a library of feces, but you're taking the DNA out. You're using subtractive hybridization or metagenomics techniques. You're using some method to say, this feces has these components that is different from the components of this feces. You're proposing that you have a marker that is going to be associated with, for instance, human fecal material. You're going to test that proposal, that hypothesis that this marker is associated with human fecal material. And when you go to the environment, you're going to extract the DNA, the whole DNA from the environment, and you're going to search for that marker. That is going to help you know, is there presence of this fecal source in my waterway? The challenge with this methodology is, of course, interpretation. What does this mean to E. coli? I don't know. How much? I don't know. So we'll talk about how to overcome some of those issues. Damn it. Early work, again, 2000, Kate Field out of Oregon. Runs in, were you on this project, Oren? Okay. You're not that old?
I was there when they did.
Okay. They did an analysis. They proposed some markers that looked like they were associated with humans. There's a cow cluster up there. And they put out some DNA probes, some primers that can be used to run PCR and probe to evaluate whether that marker is present in an environmental sample. So instead of this complex pattern that you have to interpret, I've got presence of fecal contamination, I've got presence of human-associated contamination, fecal contamination, and in fact, by doing the dilution to extinction, I have an idea of the relative amount. Quantitative PCR means that you don't have to do the dilution anymore. You can get an idea of the relative amount. You still don't know what's the relation between the marker and the E. coli and the fecal material. You still don't know whether more marker from human feces means that there is more human feces relative to another source. But you have some idea of what fecal sources are in that water. Have I bored anybody yet? Are we doing okay? Feels like it's been a long time. I've been talking for half an hour here. Has anybody got questions so far? Yes, sir.
A question on that slide with the quadrants. And you had...
This one?
Yeah.
Yes, sir.
Does that mean that the humans, you're not going to see any crossover between them and, say, pigs?
In this data set, yes. In other data sets, that idea did not hold true. And I'll show you some of the work that I did to try to do similar things in somewhere. I think it was in Nebraska. And I do not get this nice tight clustering. But in this data set, it clustered so nicely.
Okay, but it didn't hold true.
The term that Jody coined a while back was promiscuous. That as you collect isolates from multiple hosts, sometimes you find identical isolates in both human sources and especially dog sources, because dogs are filthy creatures, and pig sources. But you do find promiscuous E. coli that are genetically indistinguishable by those methods, genetically or phenotypically indistinguishable, but are found in multiple horse hosts. And if they're indistinguishable, no statistics are going to help you separate those.
And on the DNA, essentially, and that comes up quite a bit, why aren't we DNA testing? If I understand you right, it's just going to show that it could be human, could be pig, probably is pig, probably is human. But it really doesn't give us anything other that's useful beyond that.
Yeah. Well, you're jumping the gun a little bit, and we're going to spend a whole day talking about that. But I'll go ahead and answer the question directly. The question is whether or not, if you find the genetic marker for one source, what does that tell you? It tells you that source is probably there. What else can it tell you? It can't. Well, I think it can. But at face value, it's not going to tell you how many E. coli are there from that source. The question that you posed was something that triggered a response in me, which is you have got to formulate your question well. And is the fecal contamination from humans in this water? The answer is probably yes, that some of the fecal contamination is from humans. The question is an unacceptable amount of fecal contamination from humans in this water? And that's the rub. If you bring your limit of detection down low enough, you can detect human source fecal contamination at levels that are related to something like 100 E. coli. All of the regulations say that 100 E. coli is okay. So you're running a risk of correctly identifying human sources, but incorrectly identifying that it's an unacceptable level. And if you have 10,000 E. coli in your waterway and you detect a human source and you think that all 10,000 came from humans, you're going to spend a lot of money addressing human sources and only correcting 100 E. coli's worth of the issue. Do you follow?
Yep, got
it. Did that answer? Yes, sir.
Those tight clusters that you have on that graphic there, you have to do that for every study you do because do those move around? Or is a chicken here the same as a chicken in England, the same as a chicken in China?
Yes and no. Yes, you do need to. It would be first. It would be. I'm trying to think of non-negative things to say. You really ought to calibrate your methodology in your study area. If you don't calibrate, you don't know whether it's true. You've got to address your base assumptions. And one of your base assumptions is that this data set, which probably came from Minnesota, is going to work in Texas. Until you test it, you don't know whether these classification rules are appropriate for your setting. George, somebody today, I'm sure, is going to talk about regional relevance of libraries. Is that true?
Yes.
Awesome. So we will be talking about that. The shorter answer to your question is that the more isolates you add to that calibration data set, the more data you have, so the classification rules change, and sometimes you start to get overlap. And so yes, they do shift. This was, I counted them one time, I think it was 20 to 40 isolates per grouping. When you've got 20 to 40 isolates per grouping and none of them is identical, well, then you get these nice clusters. And I think I'll be showing you in just a few slides an example of 100 isolates, which is still a very small library, and what that did to my clusters. Because
Yeah, I think your horse and human example is coming up.
yes. Well, let's go there, shall we?
Oh, don't do that slide.
Okay. How about that one?
I'm doing that too.
Okay. There's the horse and human example. My point is I'm developing an argument that there are some warts. It would be awesome if everything worked just the way we want it to. It would be awesome if the ratio of E. coli to marker was 10 to 1 in every fecal source, and every fecal source had the same concentration of E. coli per gram dry mass. Everything was on an equal basis. The clusters for marker-based, for library-dependent methods, it'd be awesome if the clusters were nice and tight and isolated. And as we go through statistical validation and validation methodologies that Jody will talk about, the more data you collect, the more exceptions to the rules you find. And what do we do with those exceptions? Because we don't want to falsely incriminate one source, one stakeholder, one taxpayer we want our results to be based on hard, good science. Yes, ma'am?
You were talking that the genetic marker will tell you the fecal source, but not tell you how much. Was there then a method that you were saying would also tell you how much?
Yeah.
And I missed it.
Okay, it's quantitative PCR, and both Oren and I will be talking about that this afternoon in the session from 1 to 4, 1 to 5. I'll be talking about some of the statistical methods that Jody doesn't want me to talk about yet, and Oren will talk about some of the quantifications. So yes, you just have to be able to interpret your results, and by quantifying the marker and relating them to the E. coli, there's a chance, there's a path that can be taken to do some quantitative. Does that answer your question, sir? Okay. Other methods comparisons. So in 2000, we thought we had it licked. We were George Bush on the aircraft carrier. Woohoo! Life is good. And then Jody, I forgot to put Jody's paper in here. I did some work. The folks out in Europe did some work. The SQUIRP did some work. And just to give an overview of what the results were, when the calibration data sets came out awesome. We got great publications. We got great show of promise that these methods are going to be able to tell us the difference, and maybe even quantitatively. The calibration data sets rocked. When the researchers were challenged with blind samples of poop soup, were they able to tell, one, what's in there, and two, if there was a mixture, which one was dominant? Because I think those are two key factors. The library-dependent methods suffer from a low-level false positive detection rate. Every source, to generalize, every source was found in every sample up to a level of about 5, 10, 15%. There's noise. So unless you establish a threshold of credibility, you're just going to say, every sample comes in, I can write the report today. Every source is in that sample because there will be one promiscuous E. coli in that sample that's going to blow it for everybody, and it's going to be hard to tell what's what. The library-independent methods, the marker-based methods, tended not to be false positives, but we did get false negatives because of detection limit issues. So they both had their weak points, and none of them was able to correctly identify what's the major source in this water. I don't remember what the exact mixes were. I think the goal was 99-1. And with a 100-to-1 ratio of one source to another source, none of the methods was able to say, yeah, you know what, that source is in there, but it's not a big deal. What you really need to worry about is the second source. But again, this is 2003. This is early on. We've made advances since then. But it was enough to make everybody stop short and say, let's go back and try to refine these methodologies. And then there's the plot, if anybody cares to look at it. It's too complex to go over in the next 2 minutes. So that led to some evolution in our methodologies. You'll see words like tiered approach and toolbox approach, where instead of saying, this is my forensic evidence that the E. coli came from humans, if we scale it back and say, I have a substantial body of circumstantial evidence that says that humans are probably a major contributor, that's valuable information. That can be gained from the methods as they currently stood. There was a migration to standardized protocols. Academics, every one of them wants to do it their own way. When it comes time to standardize protocols and try to merge libraries across the Gulf states or from Minnesota to Texas, it's not going to happen unless everybody uses standardized libraries. And when the critique, one of the critiques is that the more samples you add to the library, the fuzzier the results get. You have to test what is the effect of large libraries to ensure the credibility of your data and to weed out, one of the methodologies is to weed out the promiscuous organisms. How do you know which E. coli are the promiscuous ones if you've only got one example in your library? You need 10 examples that are coming from 5 different sources to say, you know what, that particular pattern is not reliable. So I think those are the major trends in library-dependent methods. The use of not a single method solo, but to use it in either a tiered or a toolbox approach, and the drive to do standardization and consolidation. Markers, not immune. Sensitivity and specificity, there are false positives and false negatives. Sensitivity in terms of limit of detection. If you've got just a little bit of fecal material in there, or if you have a lot of E. coli but very little marker, you may not see it because of that limit of detection. The interpretation paradigm, we've probably beat that horse till it's dead and bloody. But how do you draw the link between Bacteroides or any of the other markers and the level of E. coli, which is something that by current regulatory framework, you've got to be able to link it to E. coli. Host specificity, Jody's going to talk about that. I'm going to talk about it later. We do not have perfect sensitivity and specificity. The detection of a marker is not perfect. We talked a little bit about detection of marker when the E. coli concentration or the fecal coliform concentration is actually acceptable. That's not an analytical false positive, but it is a regulatory false positive. That's something to be concerned about. Library-independent methods, the marker methods have migrated, as with the libraries, to toolbox and tiered approaches. Use of quantification, the quantitative PCR, statistical evaluation of the data instead of taking it at face value, and then the addition of additional markers. One thing I neglected to put on here was attempts to directly correlate the alternate indicators, the markers themselves, to public health outcomes. And as far as I know, the only one has been that Lake Erie study, well, the Great Lakes study, in which there was a correlation between BAC32, the general Bacteroides marker, and public health outcomes. So skip the E. coli, skip the middleman, consider using the alternate indicator in the place of the traditional fecal indicator. Future directions are going to be discussed a great deal in the coming day. I'll throw up my 2 cents worth on this slide. That there is demonstrated value to the fecal indicator regulatory paradigm. That there are flaws in both that paradigm and the paradigm that, in 2000, think of us in 2000 with microbial source tracking being similar to what the dudes were in 18. Now, painful. We've got something we can measure, but it took from 1900 to 2006 to pass the Beach Act. And it's going to take a little while for us to refine the methodologies that we've developed for microbial source tracking, but we've got a lot, in the midst of the warts, we've got a nice clean complexion in some areas that we're able to make useful interpretations, deliver useful data to users. The other thing is that different people in this room may have different needs. The regulator, particularly. relative to the pathogens of concern. And to do things like, what are some of the methodologies? Raman spectroscopy, or direct metagenomics, or microarrays. You're analyzing small quantities. You've got matrix interference. A big issue is, are you able to detect that pathogen at a level that, at its threshold of public health relevance? And I don't think that has been overcome yet. But yeah, especially when you look at the MMWR statistics and you see that the vast majority of the identified etiological agents and 75% of the diseases overall are caused by a half a dozen or a dozen organisms, well, you've got something there. And I know the Camera group up at Michigan has made some advances, CAMRA, has made some advances on a universal microarray to detect pathogen sequences. But they again suffer from matrix interferences and detection limits. All right, thank you.
Thank you very much, Don.
So our next speaker is Dr. Jody Harwood. She's an environmental microbiologist and professor with the University of South Florida in Tampa. And Jody, she's going to talk to us about the ABCs of BSD.
Well, thanks everybody for coming on this slightly gray and rainy day. I'm going to hone in a little more closely on some of the data that's led us to some of the observations and conclusions that Don talked about. And do feel free to stop me at any time with questions, because that doesn't bother me at all, and that's what we really want to get at here. And I'll make a little plug, actually. I should have put a picture of it up here, but if you're interested in a compilation of current information on microbial source tracking, Chuck Hagedorn, Anisette Blanche, and I have co-edited a book called Microbial Source Tracking, published by Springer Press. So if you're intrigued, want to know more, and have it all in one volume, that's a good source. So, let's point it at the right thing here.
Yeah, use the second long slide.
There we go. All right, so all of you are aware that water quality is a crucial aspect of the quality of our life and the length of our life. And we use water obviously for drinking water purposes. We use it for recreation, and it's important to protect it from incursions of pathogenic bacteria. And Don gave a really good introduction to the fecal indicator bacteria, how they're used and why, and that they are surrogates for the presence of pathogens. And of course, pathogens and indicator bacteria can enter our waters, our surface waters and groundwater, from many different potential sources and impact people who are using this water for all of the different uses. So that brings us to microbial source tracking in short order. And MST, my definition of MST would be using the specific or the host-associated species or types of microorganisms that are, again, really strongly associated with the feces of particular host animals, and using that relationship, as Don talked about, between these organisms that are strongly associated with feces and GI tracts of particular animals, and being able to utilize those organisms to trace back to organisms that are found in the water, and thus determine the source of fecal contamination in that water. And so just as a little schematic here, we would have either, whether it's an E. coli type, like an antibiotic resistance type, or a genomic fingerprint pattern, or whether it's a DNA marker, as Don talked about for the library independent methods, then we would isolate or purify them from the feces of a given animal type, and then be able to trace those feces back to the influence of that particular animal type in the watershed and on the water body. So that is our microbial source tracking in a nutshell. So library-dependent methods, as Don talked about, were among the first to be really explored in depth, and so that was the work of Mark Tamplin and Selena Parveen and Bruce Wiggins and Chuck Hagedorn, and I kind of came in on it at about just after Chuck, Bruce, and Mark were publishing some of their library-dependent results, and it looked really, really, really promising at the time. And still are some applications that I believe are very useful. But we would then, so the basic approach is, and many of you know this already, so if you're a professor like me, you send your graduate students out in the field and you collect fecal samples from many different types of animals, and then you isolate the indicator bacteria that you're interested in using. And we worked both with fecal coliforms, E. coli, and with Enterococci as our indicator bacteria of choice. And then you would use some sort of the ability to generate a pattern for each of these isolates. And it could be by a phenotypic method like antibiotic resistance, or it could be by a genotypic method like ERIC, rep-PCR, or ribotyping as George uses. And so having the knowledge then of these fingerprints, as Don mentioned, then you would use generally statistical methods to group those fingerprints into categories, and then being able to use a classification rule to say, well, if you see this particular fingerprint right here, then it probably came from, let's say, a dog source. And then, of course, you would collect, water from a surface water body or groundwater, and then isolate bacteria from that. And then generating the same types of patterns, you would ask the question, well, given that I have these patterns in my water body, then what is my library going to tell me about where they came from? So that would be the library-dependent MST approach in a nutshell. And one of the things that we worked really hard to convince users of early on, and we still work hard at convincing them, is that you have to do a lot of validation, a lot of performance assessment of these methods before you use them. And sort of in the early rush to use of these methods, that wasn't done as well as it could have been in some cases. But you have to know about the specificity of the classification rule that assigns these isolates to source categories. And so specificity meaning the frequency of misclassification of negative control isolates. So if you have, if you're looking at, let's say, the specificity of a method toward a human source, then you would say, well, out of all the dog, pig, cow, and horse isolates that I have in my library, what percentage of them gets misclassified as human isolates? And so if you have high specificity for a library, that means that very few of your non-target isolates get classified